Hello, and welcome to Introduction to Bioinformatics Analysis, a course taught by Phillip, Richmond. This is Section 1-5a, Introduction to Short Read Mapping. A foreword for this presentation: do not start the section until you've watched Section 1-4. Also, I highly recommend having the cheat sheet out for this section, as I won't be walking through the basic commands. So quick start instructions. Let's go ahead and open up our terminal and log in to the Westgrid compute server. Enter in our credentials, and voila, we're in. So in this presentation, I'm going to be going over mapping of short reads back to a reference genome. So in order to kind of set a base of knowledge for where these short reads are coming from, I'll quickly walk through the Illumina DNA sequencing technology. So genomic DNA is sheared and then size selected, and you ligate adapters to your pieces of DNA, attach them to a flow cell, and then there will be a cluster generation where you essentially run through bridge amplification and generate these small clusters of identical short DNA sequences. Then each of these clusters, essentially on the flow cell, are washed over with the dideoxy NTPs, your A's, T's, G's, and C's, and you wash the entire flow cell with all of them, and they are labeled with a fluorescent tag. And then as soon as they attach, they are excited with a laser, and whichever base attached will emit a certain fluorescence reading for that cluster. And then these essentially images are deconvoluted, and you can get essentially a readout for every single cluster on the flow cell, every single position within what we ended up having are reads. So these reads are also accompanied by a quality score, and I will go over that quality score in a minute, and they are contained in what we like to call a FASTQ file. So a FASTQ file will have the extension .fastq or .fq, and each read within this file are represented by 4 lines. So first you have @ and then the read name, which usually is going to have something to do about the flow cell information for Illumina sequencing. Then you have the actual ATG sequence, and then you have the a line that is just pretty much usually a plus sign. Sometimes there's repetition of the read name here, and then you have an ASCII encoded quality value sequence. And so these quality values are ASCII encoded so that you can have a larger range of scores represented by a single digit. Whereas if you had just numerical scores, as soon as you got from 0 to 9, you would run out of possible scores and your spectrum wouldn't be as large. So let's go ahead and take a look at our own FASTQ file. The toy data set that we will be using in this presentation comes from the Lambda virus, which is an enterobacteriophage. The genome is not too large, and that's why it is a test data set. When we run the commands and these different mapping operations, it will go very, very quickly. This scales, the time scales much longer as you have more complex and larger genomes and larger data sets. So let's go ahead and copy all of these files into our sandbox. So first of all, let's just go to our Wine Research Center directory, Global Scratch Wine Research WRC. Do an ls. We can see that we have our sandbox directory. So once you're in your sandbox directory, what we're going to want to do is we're going to want to copy from /global scratch WRC data raw, and we're just going to do all of the .fq files, and we can do that by typing the asterisk or star fq, and if we hit tab twice, you can see that I have listed all of the reads. So this star says any file, doesn't matter what it's called, as long as it ends in fq. So you can see all three of these files end in fq, and we want to copy that just right to where we are. So if we do an ls, we can now see that we have these reads. I had them before, so that's why they're there in this ls and that one. Next, we want to copy the genome.fasta file. So again, cp/global scratch wrc. This one is in genomes, lambda virus.fa to our current directory, and again, there it is. We can see that these files were just copied here. And let's take a look at the reads_1 fastq file using more, but I'm actually going to use less. So less on reads_1.fq. And so you can see, again, this is a toy data set, so there's no flow cell information here, but you have @ and then the name of read 1, and then the sequence of read 1, just a plus sign, and then the ASCII encoded quality scores for read 1. Next we have @read 2, so this is 1, 2, 3, 4 lines, and then 1, 2, 3, 4 lines. So as these lines get longer, you can see they have to wrap, but that's the nice part about less, is that as we scroll out like this, or if we were to shrink, we could really see that these are 4 lines per read. And I'll go ahead and make that bigger again so we can see it and use it. Fix the size a little bit, and we're going to get out of less by typing Q, and then we'll do a clear. Okay, so we've got our files and we've looked at our fastq file. So the tool that we're going to be mapping with is called Bowtie2. Now, these mapping algorithms, I highly recommend reading the references and the papers that accompany these different tools. If you are interested in talking more about these algorithms, then I'm happy to do that with you offline, but for the purposes of this course, really what you need to understand is that Bowtie2 is going to use a compression-based algorithm in order to transform the genome, which is a long linear string, into more of a graph, that when we map to this graph, it's actually a suffix tree, we can leverage certain algorithms in order to speed up the process instead of having to scan the entire genome. So from a matter of execution, the first thing we need to do when we use Bowtie2 is we need to generate an index of our genome.fasta file. And so we use the Bowtie2-build command for this. So whenever we want to use a program or a tool for the first time, we can call it in the same way that we call upon a command line command. And so that is just by referencing the location of the tool. And then adding a -h will usually get you some help options. So this tool, we'll type /global scratch wrc. I've installed it in the tools directory under Bowtie2. Remember, tab complete is your friend. Bowtie2-build /h. And you can see when we have that -h, it prints out the usage statement for this tool. So you can see there are a number of options, but being able to interpret this usage statement is what is going to make using new tools much, much easier for you. So let's break down this usage statement. We have this line here is really what is required. So Bowtie2-build, the brackets signify optional arguments. So this is where we would put our options. We would put them right after the Bowtie2-build. put all of the options that we need. In our case, we're not going to have any extra options. We're just going to use the default. Then followed by a space, and then you've seen me use these before, the greater than, less than. Again, they just want you to substitute what's in this space with a reference sequence, and then followed by another space, and then followed by the output name that we want these indexes to be called. And so down here you can see my full command, and I will type it in here. So we will type, again, if we just hit up, we can go right back to that command, and we want to do that on the lambdavirus.fa. And again, I can do that like this because this actual FASTA file is in the directory that I'm currently in. I'm in my sandbox directory. And then I want to call it something like lambda virus bowtie 2 index. Something that you may notice that just happened there is that my command started to wrap around. So sometimes what we can do in order to prevent that is we can stretch our screen a little bit, and that way the commands won't wrap. Or if we shrink our screen, this is also going to allow us to avoid that wrapping. So this is the full command here. As these commands get longer, I will use smaller font so that you can see them a little easier, but again, all of the commands are also within the slides. So you can see a lot of things printed to the screen when we ran this. Most of this is just essentially nonsense information about the status of it. But if we run it again, you can see it runs very quickly. So now if we type ls, you can see that what we've done is we've created these .bt2 or .bowtie2 index files. So there you can see there are 6 of them here created. The next thing we're going to want to do is we're going to want to map our reads.fastq back to that reference genome. And so we're going to, again, using a tool for the first time, we are going to run just the tool with the -h command to figure out how we use it. Now, you can see when I use the -h command, this has many different options available. However, again, if we come up to the top, these are the only required options these are the only required parameters with these optional arguments built in at the beginning. But breaking down the required arguments, we have -x, the location of our bowtie2 index that we just built, and then -1, our reads_1.fq, -2, our reads_2.fq, these are paired reads, and then -s sam. Now this is optional, but this is the best way to specify our output file. So again, you can see my full command here, and we can type that in. bowtie2-x, the location of our index. It's good to use full file path. I'm not going to for the purposes of this example because I know exactly where this index is, and so when we refer to the index, we refer to it all the way up to that period. So don't use the period in your index when you are listing it with bowtie2. Then we have -1, our reads1.fq, -2, reads2.fq, and we'll call this lambda bowtie2.sam is going to be our out file, and we need to specify -s. So when we hit enter, there were no errors, and voila, just like that, we mapped our 10,000 pairs of reads back to the genome. So you can see that something was printed out to the screen, and what this is essentially the standard error. So unlike the standard out, the standard error can be captured by adding a 2 greater than to the end of your command, and we want to capture that output in a file called, let's call it lambda bowtie2.stderr, short for standard error. And now when we run the command, you can see nothing printed to the screen, but this way we can save off our mapping statistics. So if we take a look at this file literally just has essentially what was printed to the screen there. You can see that we had a 94.97 overall alignment rate. There are a number of reads that did not map at all, so those probably make up some sequence that was not in our reference genome. So looking at the output of this, I'm just going to clear the screen here. Let's go ahead and take a look at our SAM file, or standard alignment file. So a typical standard alignment file is going to start with these at lines. So at SQ is going to be essentially the reference that we map to. So there'll be one of these for every chromosome. Again, this lambda virus only has one circular piece of DNA that is 48,502 bases long. So it's very short. And then you have some more information here about the command that we actually ran. And what's nice about this is that it will actually tell us all of the options. So you can see we didn't really use any special options, and so what's printed here is just the default that we ran with. But if we perhaps ran with a number of different options about controlling the output or controlling the amount of mismatches allowed per read, this is something that we'll get into next week, that would appear here. And then we get into our tab delimited read by read output. So you can see these first two paired reads here. Each one has its own line, but they're both called R1. Anyways, we can see that, let's just break down one of these lines, and the easiest way to do that is to go to this README here, which details the SAM format. Just walking through these fields, the first column is going to be the name of the read or the query name. The second column is going to be a bitwise flag, which we won't have to worry about for now. The third column is going to be the chromosome or the reference sequence that it mapped to. POS is going to be the position that that read mapped to. So you can see this mapped to position 18,401. The mapping quality, 42 is a perfect quality. You can look at the different mapping quality options either in the Bowtie 2 manual or on Wikipedia. And then some more information that you can go through on your own, including a cigar string which details any mismatches or insertions or deletions, which is represented here. So there are no insertions or deletions with the equal sign. Perhaps there are others elsewhere. Anyways, that's something we can look at a little bit later. So let's go ahead and quit out of that. That was our SAM file. We're going to be dealing with a lot of SAM files, so it's very important that you're comfortable with this file format. However, the downstream tools that we're going to be using on our alignment files are not going to want to read a text file. Instead, they're going to want to use as input a binary file, a compressed file. And so what we can do is we can compress and create a binary alignment file, a BAM, using the SAM Tools suite. And so the SAM Tools suite will do a number of things. The first thing we're going to do is we're going to convert from SAM to BAM using samtools view. So samtools is located in /global scratch wrc tools samtools. So when we type samtools, you can see there are many different options that we have for files. The one that we're going to start with is going to be samtools view, because this is going to convert our file formats. So samtools, oops, we'll just go back to what we had, view, and if we type samtools view, now that we've invoked a specific tool, we're going to get a specific usage statement. So samtools view, a number of options, some inputs, and I will just go ahead and break this down. We're going to use options -lowercase b, which means that we want an output BAM, and -uppercase s, meaning that the input is SAM, although I guess this is now ignored, and then -o for our output.bam. So samtools view -bs, then our input, which is lambda virus or lambda bowtie2.sam, and our output, which we will call the same thing, .bam instead of .sam. And just like that, now you can see we have our BAM file, which we can take a look at. However, if we try and take a look at our BAM file, we're going to get some weird things printed to the screen. Some odd characters. It's going to be very confused because this is not what we call human readable. It's binary. It's only machine readable. So now that we have our binary format, we're going to want to sort it so that it's sorted by read position. So we can use now the samtools sort command. I'm going to go ahead and erase this. Samtools sort. And again, if I just type samtools sort, it's going to give me the options. So let's go ahead and use samtools sort. Our input is going to be, all that we need to do, I guess, is provide the input, and then let's also provide an output instead of writing the standard out. So samtools sort, our input this time is going to be lambda bowtie2.bam, and our -o output is going to be lambda bowtie2.sorted.bam. So you can see I'm going to just add this .sorted to the file name, and that's going to let me know that this file is sorted. So now I have my SAM, my BAM, and my sorted.bam. Lastly, we're going to want to create an index of our sorted BAM file, and this is required by several different tools, including when we visualize the data in the next section. So we're going to do this using samtools index. So again, it's easy just to scroll up here. We're going to samtools index, and if we just run it without anything, you can see very easy. Only thing that we're going to want to do here is just have the in.bam. So samtools index lambda bowtie2.sorted.bam, and just like that, it produced the index file for that sorted BAM file. Now lastly, what I want you to do, because writing out all of these commands several times can get very annoying, to say the least. So one thing that we can do is we can save off all of our commands in a shell script. So what we'll do is we'll save off, essentially what we've built is a pipeline, an analysis pipeline, and put it into a single shell script. So we can copy command paste that worked from the shell into a script using Emacs, and then I'll show you how to submit that script. So generally when I'm doing this, I like to have two different terminals open. One terminal where I am going to be typing things, and the other terminal where I will be copying commands. So I will go ahead and log in. And you can see I can have multiple terminals open with being logged into the same compute server. So let's go ahead and change into our sandbox directory, global scratch wrc Richmond, and I'm going to type Emacs map and convert.sh. So I am now in my file editor, and this was a command that I ran that worked, so I can just copy paste that, and voila. Now I will copy paste the following commands, which included a samtools view, if we keep scrolling through, we can now do a find our samtools sort command. And then lastly, our samtools index command. Now again, to save, Control X, Control C, and we'll hit yes. So now if we type ls, we now have this map and convert shell script.sh file. And so we can submit this using sh map and convert.sh. So one thing that I'll do here is just real quick, we'll do a clear. We'll do ls-lahtr. This is my favorite one because it shows you in a reverse order R, sorted based on time, human readable, and long format. And the A is just showing me hidden files as well. So we can see that these files were all generated between 1622 and 1630. And now if I type sh map and convert, it's going to go through each of those commands and execute them. And now, because it finished without any errors, I can see that all of these files were updated and they were just now produced at 1634. So that is one of the beauties of the shell scripts, is that it will show us, one, exactly how we ran our different commands, but now if we wanted to run this on a different input set, we could just copy this map and convert file and alter a few file names within this without having to retype all of the commands every time. That's all I have for this presentation. Goodbye.